All right, so our last speaker today for this session is from one of the beneficiary organizations. It's with us Dr. Enrico Guarnera from Merck, who is going to talk about from statistical mechanical models of protein function modulation towards the design of allosteric drugs. Please.
Thank you, Giovanni. So first of all, thank you so much for the invitation. This is really a privilege, I have to say, also to listen to Professor Chen lecture today. And also, for me, the first time to be in Urbino, the city of Raffaello, and so which we learn in school, and it's really nice. So today, let's say I'm the last one today, so you're gonna be patient with the simplicity of my talk. So I will go to the very bottom of the models that wants to, let's say, show some predictivity in Allostery. So I like to think as Allostery as a sort of mindset in drug discovery and development. And so when you think in those terms, you start from, of course, the traditional definition of Allostery, meaning, let's say, regulation of protein activity as a result of effector. However, when we look at, let's say, pictures like this in cellular networks, where we see all these proteins, let's say, in contact between them, receptors and the nucleus, somehow one has the impression that the definition of Allostery should be kind of generalized. And I like to think in a sort of comprehensive definition, where, let's say, modulation of the protein activity is really the result of a perturbation in very in a generalized terms, where not only effector binding is considered, but also mutations, but also protein-protein interaction disorder, but even pH and post-translational modifications. And I think the key to, let's say, to think about those things is energy landscape perspective. When you have let's say, a profile like this, where, let's say, your system is unfolded here and here it's folded, and then you have this ruggedness, which in principle would correspond through, let's say, the effect of perturbation to a modulation, let's say, as a result of mutations or effect of binding or etc. One asks ourselves, let's say, what is the minimalistic model that might, let's say, give some description and, let's say, how we can learn from it. And so, and I think, so on this respect, let's say, communication, what is really important in allosteric modulation? What is driving this this communication? And most important, I think, how we can really identify potential allosteric sites. Let's say, it's important, of course, that one has to, let's say, to think in a medchem perspective, identifying ligands, and I'm part of a pharmaceutical company which is obsessed with these things, but I will not talk about this. In fact, let's say I'm part of Merck, but I will not tell you any secret of Merck. I will talk about work that I did in academia until some time ago. And so let's say, a minimalistic model of Allostery should really take into account, let's say, an energy landscape perspective, but what I think very important is causality. So causality, what is really driving perturbation, what perturbation can let's say, how perturbation can act on the system. And I'm a physicist. I think in terms of energetics, and when I think in terms of perturbation and their effect, I think in terms of what happens to the system and how degrees of freedom are modified, how the energetics is modified, and how we can link this to modulation. And let's say to think in those terms, we came up some time ago with a minimalistic structure-based, we call it structure-based statistical mechanical model of Allostery, which is talked about coarse-graining today. It's by definition, let's say, it's intimately a coarse-grained harmonic model of protein Allostery, which has the aim to simulate ligand binding, but also mutations in a quite generalized sense. And the point is to provide a residue description of allostery and quantifying let's say, their effects in terms of energetics. And ultimately, what the output of the model is to provide a delta G, where the delta G is the result, let's say, the response at the single residue level as a result of a perturbation. So the model is as, let's say, three components very quickly. In a sense, it's as I was saying, it's an harmonic model where in a sense, we consider two systems, let's say, two states. The protein in its, let's say, free, unbound, unperturbed state, and versus a bound system where let's say, if we consider binding, ligand binding, we apply a restraining, an harmonic restraining on the binding sites. Or if we consider, and this would be the energy function associated here. So this is a term that is restraining, harmonic restraining with respect to the, let's say, the unperturbed energy term. But there are also, we can consider also a mutated state where mutations essentially are of two types, either mutations that tends to stabilize, increase, let's say, the interactions on the local network, or destabilize, meaning that, let's say, the network of interactions is depleted. And of course, you can actually combine the two things. How you consider, let's say, the allosteric effects? Essentially, you consider let's say, you define investigate the conformational ensemble of this in the space of normal modes, where, in a sense, you generate all possible configurations starting from, let's say, a starting structure that comes from the PDB. And so this is a little bit equivalent to a diffusion model that we very recently let's say, we are flooded with. But let's say, as a result of this superimposition of harmonic normal modes, essentially you define a state. So each conformer here can be uniquely defined by a vector of parameters, and so with that you can define a potential that we call it, let's say, an allosteric potential, which nothing else does than evaluating the elastic work one residue is performing to resist, to counteract the motion of its neighbors in space. But then, what you can do once you have such a model, you can really calculate partition functions, and so that would be really integrating over those vectors of essentially diffusion parameters. And once you integrate it over those, you're going to end up with a free energy response at the single residue level, which is of this type. Then, of course, you can evaluate things per site. You can average over the sites, but then you can calculate things like what we call allosteric modulation, which essentially would clear up the effect of the mutation, let's say, of the perturbation out of the background effect. That is to distinguish the response due to a perturbation that is not allosteric from a perturbation that is acting globally on the system. For example, for mutation, we define what we call allosteric modulation range, where essentially we consider the maximal extent of the perturbation in terms of the energy response from, let's say, mutation that tends to destabilize with respect to another mutation that stabilizes. And typically, this is the type of results that you get. This is a profile where this is PFK, phosphatokinase, so one of the most famous systems, allosteric, classical allosteric systems. And so the residue here I'm averaging over the chains, and here I'm showing essentially the response of the delta G's as a result of binding at one of the ADP allosteric sites. And when you alternatively use mutations, for example, here I'm applying mutations in one of the domains of the chains, you end up with a distribution of responses that tells you where this, let's say, what is the answer, what is the response over the system. And so we applied this on a number of, let's say, classical systems. Again, here this is the phosphofructokinase. Here so phosphofructokinase has this very two different behaviors due to the different inactivator and inhibitor. There is also the presence of this ADP functional working in combination with the catalytic site. And you see here, here I'm showing you the results for binding in the PEP site. You see this is the region where this is the binding taking place. Here, these are the positions of the allosteric site. In blue, you have those regions with the delta G that is positive, which you can fairly interpret as active work being exerted locally, whereas in red, when it's negative, so that essentially would be stabilizing work. So when it's positive, essentially you can consider that region as prone to conformation change, whereas when it's negative, it is less prone to conformation change. We applied this, as I said, on a classical set of allosteric proteins. So here I'm showing you the ATCase, which is a beautiful, everyone of you knows, beautiful heteroimeric giant enzyme, which nicely shows here. These are the locations essentially of the allosteric sites. And in a sense, when those allosteric sites are bound, there is the functional region of the protein gets, let's say, highlighted, essentially suggesting that there is active work being performed at that site. Yeah, these are other systems. Actually, this is not a malignant hydrogenase. This is another system. But let me go fast. So we understood that essentially allosteric communication is the result of a sort of redistribution of the active work in the protein as a result of a perturbation at the allosteric site. But one can ask ourselves, okay, what about the other way around? Can active sites actually, upon perturbation, communicate with their allosteric site? And so we asked this question, whether a sort of reverse perturbation can be considered, meaning that if you, instead of perturbing allosteric site, you do perturbation at the catalytic site, what happens to the allosteric site? And so we applied this on the same data set, on the same, let's say, protein set that we had, and with some, let's say, a fairly, let's say, predictivity we could achieve in terms of the known allosteric site. So these are some predictivity data associated with also another data set that we use, the ESA bench from the University of Shanghai. However, with the, let's say, with the predicting allosteric site, the business is a little bit tricky because allosteric site, let's say, if allostery is an intrinsic property of proteins in terms of, say, their dynamics as a result of perturbation, annotation is a limitation. So what I'm saying is that in the prediction, a fairly high number of false positives can emerge. But in many cases, those false positives could be potentially active allosteric site. An example is the bovine glutamate dehydrogenase enzyme, which is an extremely complex enzyme, hexameric, that has allosteric sites fairly close together and not that far from the active site, but still allosteric. And so in this case, this is one of the typical situations where the simplicity of the model, of course, it's tricking us. However, we can identify quite astonishingly well this antenna region of the enzyme, which is a bundle of 3 helices, which has been known to play a crucial role in the cooperativity of the allosteric function of this enzyme. At the same time, when you say this, why this let's say this approach of generating a potential allosteric site, you can actually generate multiple allosteric potential allosteric site. Here I'm showing you the NAP-dependent Merck enzyme. Here these are 5 potential nodal allosteric site, and with let's say with some energetics associated with them. But the cool thing is, you can ask yourself, okay, what if I change the residue composition of the site, and how does it change the response, the allosteric response over in the problem? Okay, so since we were having fun doing all this, we went over, let's say, we made some next steps. And since the model is intrinsically simple and allows to do many things, so we defined 2 types of objects to, let's say, to introduce, let's say, along the direction of defining what are the allosteric fingerprints associated with protein folds. And we defined two different objects. One is the allosteric signaling map, in which essentially we exhaustively apply mutations or perturbations, single point perturbations all over the system, and with responding, so that we can see responding residue, and you see here, this is applied on PFK again. You see here let's say of course the multi-chain structure, but also the domain structure and the different type of let's say positively modulated versus the negatively modulated. But we will also consider another object, which is the allosteric probing maps, in which 3 Merck probes were used to essentially probing binding, scanning all over the protein and identifying what are the regions that might potentially lead to yeah, let's say to modulation in the catalytic site. With that, with such a tool, among the other things we did, we investigated the analysis, let's say, the allosteric signaling on protons and on protein folds, and of course, modern proteins are the result of approximately 3 billion of evolution, years of evolution, and which are the result of a hierarchy of, let's say, loop structures, domain structures, quaternary structures. And so it is a fair question to, let's say, to ask ourselves whether some fingerprints exist across the different protein folds families. Here I'm showing you just one example, which is allosteric communications on the beta propellers, which yeah, here we exactly. So here we consider the allosteric modulation, the allosteric signaling map. So applying all possible perturbations, single point, and we could intersecting those perturbations with the binding site, we could see some directionality and specificity in the way signaling is organized across the protein folds. I cannot go into details here because we will stay until tomorrow. Then let's say, okay, we hope to forget about COVID, but since this is an interesting story that from us, one of the natural systems we wanted to analyze was the spike protein. And actually, one of the questions that we wanted to answer, we wanted to ask was whether we could identify some, let's say, what was the role of variant of concerns, and whether allosteric effects of mutations could play a role. What was the role in association with the variant of concern? And that, but also the role of glycosylation. Giorgio was mentioning about this today. In particular, yeah, here I'm showing you a collection of the role of the mutations associated with the variants, let's say, let's say, the development of the different variants across time, and how they played, let's say, what was their role, what is their role in modulating the receptor binding domain of the spike protein. We're actually working right now on establishing whether there is a saturation effect in all possible. This is, let's say, an interesting topic in general in viral evolutions. How mutations in the evolution of variants, what is the maximum level of mutations that you can introduce such that you go into saturation with respect to the interactions with the targets. So this model is the model that we have been talking about up to now is encapsulated in a server which is publicly available, which is, let's say, it gives you the possibility to investigate simple, let's say, using a very simple and relatively fast a model. Let's say the phenomenology associated with perturbation and response, but also we compiled a database of different. Here, these are the proteins, allosteric proteins I was talking about, but we also comprised together allosteric polymorphism. So we analyzed the role of mutations that are associated with cancer drivers, but also more recently these two databases for Rosetta and AlphaFold. This is a great data set if you want to do machine learning, especially if you want to reconstruct make predictions on some effects of perturbations and mutations. The model I have recently formulated a different model, which is exclusively network-based, but where the information of the sequence is also encapsulated, and which takes advantage of the Miyazawa-Germinal coupling energies. And the cool thing of this model is it's purely network-based and purely statistical mechanical, and allows to essentially to decouple the enthalpic and entropic parts, which is not that trivial when you consider purely structure-based models. So I hope I give you a glance of, let's say, a conceptual model that can help a little bit understanding the physics of allosteric communication. A method also based on this reverse perturbation, which I think, if considered in a context of higher accuracy of the model, can, in my opinion, can potently identify regions that are actively allosteric, and as a result, one can design either let's say small molecules, but also antibodies to modulate the corresponding system. I also talked about the idea of tuning the response of an allosteric perturbation, and the role of mutations in the context of viral evolution. And yeah, so that concludes my talk. These are the people where this work has been done. Ivo Berezovsky, Wewen T, and Sanwa from the A-Star in Singapore. Thank you so much for your attention. Please. Yeah.
Great talk, thank you. I really enjoyed it. So you said that you have found some signature of secondary structural elements. Would you say that you group
I would say that the former. So I would say that domain structure is absolutely crucial. And in fact, actually, I didn't talk about it, but we found also that typically the allosteric response decay, let's say in a perturbation, let's say you apply perturbation on the system, typically decay on a length scale of about 35 angstrom, which is the typical length of domains. And this is actually something that's been also evaluated in chemical shift perturbation experiments. I was astonished myself to see that. That really, and more importantly, there is a sort of alternate perturbation response structure associated with the domain structure. So when you typically have a domain that is negatively responding to perturbation, the next domain typically responds positively, like a sort of compensatory. However, it really depends on the fold structure, which is why we analyzed let's say, we performed the sort of atlas analysis on protein folds. However, there is still something missing, let's say, the evolutionary aspects, which are escaping me, in particular, let's say, the sequence aspects. So domain structure. So when we talk about Allostery, we always do structure-based. When we introduce sequence, it's a little bit tricky, let's say, to identify really changes that can dramatically modify the allosteric response. And I think this is also now in context of machine learning, I think it's a great moment to investigate that. So going towards patterns, sequence patterns.
Yeah, so very nice talk. So we know that allosteric proteins, they live in multiple conformations. I guess your analysis is structure-based, so you have one structure, right?
Two. Typically two.
You take two structures?
Yeah, two structures. So let's say well, yes, I have one structure. So I have the PDB structure. But the point is that when you apply the perturbation, that's not the same structure anymore. So when you apply the perturbation, either the mutation, perturbation, let's say mutation-like perturbation, you change the network of interactions, and potentially you can also cut the interactions.
Yeah, sure. But
so and that
the topology depends on
the topology, yes.
So what I was wondering is if you have a protein, so you have an active state or inactive state of the protein with two different topologies.
Yeah.
How do you consider this in the model? Your model of Allostery, or do you consider it or not?
In the purely structure base, you wouldn't see a difference. Let's say if you wouldn't see a difference in that case. But in the sequence dependent model, yes. So in the more recent one, there we consider sequence dependence. So even if you have the same structure but different sequences, you would have two different kind of phenomenology associated with the perturbation. But that's the intrinsic limitation of the model. But the question we had was, what is the minimalistic model that through which we can say everything about the allosteric of that system, and what can we learn out of it? And we learned a number of things, which is astonishing to me.
Thank you.